Hello, my dear students. I hope you all have been doing well. Today we are going to discuss about urea cycle. This is my lecture number 13, and this urea cycle is the part of our topic that is protein metabolism. In this topic, first we studied about the how protein digest and absorb, and then we discuss about that after the digestion and absorption of protein in the form of amino acid into the microvillus cell, and how it transport to the liver, and from liver, if it is required, uh, if amino acid are required to build other protein that is needed in our body, so how the amino acid use for the anabolism of protein, and how if the and after reaching the liver, how amino acid further catabolize inside the liver cell. So this was all about that we have studied up till now, and from this lecture, in this lecture we would be studying that how um, how the ammonia group of amino acid convert into the urea. So in my previous lecture I discussed with you that amino acid, when protein digest and absorb in the microvillus cell of our intestine. So after the absorption, so first of all I wish to give you an overall idea what we how protein when come into contact with our body, it start digest and then absorb, and then what happened with it. So just keep in mind that when we take protein in our mouth, so protein is made up of amino acid and amino acid linked together via peptide bonds. So a protein, a polypeptide chain is having multiple polypeptide bond, multiple peptide bonds. So the first step of protein digestion is the breakdown of this peptide bond. For that, various protease enzyme work together and then convert the long polypeptide chain after breaking the peptide bond into smaller dipeptide and tripeptide. And then dipeptide and tripeptide release individual amino acid within our intestinal cell, and from there, after the release of um, individual amino acid um, from the long polypeptide chain or from the dipeptide and tripeptide, the individual amino acid enter in the microvillus cell. From there they transport it into the liver. So in liver, what happened that transamination and deamination of amino acid takes place. And what is the deamination? In the process of deamination, amine group of amino acid remove from the amino acid, and then this amino group convert into ammonia. So after conversion of this amine group into the ammonia, what happened, this ammonia then enter in the cycle, that is a series of biochemical reaction, and it takes place in the liver cell, liver hepatocyte. And these ammonia then after the various biochemical process convert into urea, and then this urea excrete out from the body. So this cycle, in that ammonia enter in the biochemical reaction and convert into urea. So this cycle is called as urea cycle, and we are going to discuss about this cycle. So you have to just keep in mind from where we are starting this lecture, or for what we are going to study in this lecture. We are going to study that how the ammonia that releases from amino acid after the deamination reaction, how this um, amine group that is released from the amino acid after deamination reaction. How this amine group convert into ammonia after entering a series of biochemical process and convert into urea. So this we will be studying about the urea cycle. So first of all, what is the urea cycle? Urea cycle is a cycle of biochemical reaction that convert ammonia into urea, and this cycle is also called as ornithine cycle. So why we are also knowing this cycle as an ornithine cycle, I will discuss you in coming slides. But till here you just have to keep in mind that urea cycle is a cycle of series of biochemical reaction that convert ammonia into urea, and this cycle is also called as ornithine cycle. And why this cycle is necessary to takes place, and why it is necessary for ammonia to convert into urea? Because ammonia, when amino acid catabolize, and amino acid releases is amine group, and amine group uh, convert into ammonia after different chemical reaction that takes place in our liver. So we are not discussing these reaction, but you have to just keep in your mind that amino acid deamination remove amine group from the amino acid, and this amine group after series of chemical reaction convert into ammonia. And then ammonia, why ammonia, then ammonia convert into the urea after going into the urea cycle, and then urea excrete out from our body. So question is this: why this ammonia cannot excrete from our body as in the its present form, in the ammonia form? Answer is because the ammonia is highly toxic in nature, and it is not means very much soluble, so it cannot be excrete out from the kidneys, renal tubules easily. So you know very well that to be excrete out from the kidney, renal tubule or kidney cell, a compound or a nutrient or a molecule should be very well water soluble. So ammonia is not very well water soluble in very much ammonia is not very well water soluble, and it is highly toxic. So what our body does with the ammonia after the series of biochemical reaction, bodies liver hepatocyte convert this ammonia into urea. So urea is very much water soluble or easily water soluble. So it is and it is also non-toxic in nature. Urea is also non-toxic in nature, so it easily excrete out from kidney, kidneys, renal tubules. And this was the reason why ammonia convert into urea to be excrete out from our body. So you are now taking in your mind this clear thing that what is the end product of protein metabolism. So this urea is the end product of protein metabolism. We from our very first lecture that we start discussing about the protein metabolism, so we start discussing that when we take protein in our mouth, means in the form we take it in the form of collection of polypeptide chain, and these polypeptide chain consist on a number of amino acid that are linked together with the peptide bond. So the means protein metabolism start from the digestion, and then protein in the form of single amino acid absorb after converting into single amino acid absorb in intestine, and then from intestine it reaches to the liver cell, where amino acid further catabolize, and then after catabolizing, the amino acid convert into ammonia, amine group of amino acid convert into ammonia, and then ammonia convert into urea, and then the urea excrete out from our body. So this was whole the story. Now question is this: where this as I already have discussed with you to discuss any biochemical reaction or any biochemical pathway of catabolism, of anabolism of macromolecule. You should also have to know that where these reactions are taking place in your body organ, or in what organelle or what cells they are taking place. So when we talk about the urea cycle takes place in kidneys hepatocyte, in liver hepatocyte, primarily in liver hepatocyte, but sometime or a very lesser extent, this urea cycle may also takes place, or or also takes place in kidney cells. But the main prime organ of urea cycle, uh, um, uh, prime organ where urea cycle takes place in our body, is the liver. And what organelle this urea cycle takes place? Urea cycle completed into two phases, you can say, because on the basis of their compartment where it is takes place inside the cell. So half of the urea cycle, or a small or few reactions of urea cycle takes place in the hepatocyte cytosol, and then rest of the biochemical reaction of urea cycle takes place in the mitochondria of hepatocyte, or liver or kidney cell. So you have to keep clear in your mind that urea cycle takes place both in the cytosol and then mitochondria. And you also know have to know about this, that whatever whenever we eat protein, so this whenever these protein metabolize, so approximately 90% of the whenever protein metabolize, so we are taking its end product in the form of nitrogenous molecule or compound, because amino acid containing the nitrogen atom in the form of amine group. So the from this 90% of the nitrogenous excretory products, 30 gram of these products contain ammonia. And whatever and the that ammonia convert into the urea. And the urea, how urea form with the combination of ammonia with carbon dioxide and another amino acid, that is the aspartate. So this aspartate give its nitrogen to amine group and the carbon dioxide. So these all three things form the urea. So we can see here that what are the substrate of the urea cycle and what are the end product. Means how the urea cycle start, from what substrate urea cycle start, and what product it generated. So urea cycle start from the ammonia that we that generated after the removal of amine group from ammonia from amino acid. And it is also used as a substrate, um, carbon dioxide and nitrogen atom of aspartate and 3 ATP. And what product urea cycle produce? Urea cycle end, or urea cycle produce urea, fumarate, another amino acid and water. So here this is the cycle. So we will discuss here step by step how urea cycle start and how urea cycle ends. So as I told you previously, that urea cycle start from the ammonia, that from from where ammonia generated from that amino acid that reaches in our liver after metabolism, after means releasing these amino acid from the polypeptide chain in the microvillus cell of your intestine. So microvillus cell of intestine transported single individual amino acid to the liver, and after the series of biochemical reaction that we will not discuss here, these amine group form ammonia. So what happened then with ammonia? This ammonia inside your liver hepatocyte cytosol. So inside your liver hepatocyte cytosol, this ammonia combined with carbon dioxide and form carbamoyl phosphate. So for this reaction to takes place, enzyme carbamoyl phosphate synthetase 1 help. So as the name of this enzyme carbamyl phosphate synthetase. So as the name of this enzyme is indicating that this enzyme is synthetase. Synthetase, what the synthetase enzyme do? Synthetase enzyme combine two molecule together or condense two molecule together. So what this synthetase enzyme is combining, or what molecule it is assisting for combining? It is assisting carbon dioxide uh, to combine with ammonia. So as the name carbamoyl phosphate, it indicates that this synthetase enzyme allowing carbon dioxide to combine with ammonium and form carbamoyl phosphate. So what is happening next? Next, this carbamoyl phosphate then combine with ornithine and then form citrulline. So these two reactions, condensation of carbon dioxide with ammonium with the help of enzyme carbamoyl phosphate synthetase 1, and with this result of this condensation, formation of carbamoyl phosphate, step 1, reaction number 1, and then reaction number 2. What is happening in reaction number 2? This condensation of carbamoyl phosphate with the ornithine and the formation of citrulline. So these two reaction takes place in the mitochondria, and then uh, this citrulline transferred into the cytosol, and the rest reaction takes place in the cytosol of the hepatocyte. So what happened with this citrulline we will be seeing here. Then citrulline uh, actually form after the reaction of ornithine with carbamoyl phosphate. So enzyme ornithine transcarbamylase help for this reaction, and then as a product we get citrulline. Citrulline with the help of an enzyme, that is arginosuccinate synthetase, form arginosuccinate. So again we are having here an enzyme that its name is synthetase. So as we have already discussed, if whenever we will be having enzyme synthetase, it is meaning that enzyme is combining or condensing two molecule together. So what condensation, what molecule are condensing here? Actually citrulline is condensing with aspartate. So these are two amino acid. This one is the amino acid. So what happened? This actually this amino acid aspartate nitrogen combined with the citrulline with the help of enzyme arginosuccinate synthetase, and then form arginosuccinate. This arginosuccinate then release or lyase, or you can say break down into two other molecule, that one is fumarate and other is arginine with the help of arginosuccinase. So this arginine then with the help of enzyme arginase release urea, and then release urea and ornithine, or we can say this arginosuccinase break down arginine into urea and ornithine. So whenever we are will be having enzyme name that is ending on uh, just ASE, ASE indicating that it is an enzyme. And so what is happening here? Arginase, it is means indicating that this is the enzyme that is breaking down arginine into urea and the ornithine. So what happened with this urea? The urea then enter in your blood. From your blood it reaches to the kidney, and from the kidney it reaches to your renal tubules, and from the renal tubules it is excrete out from your body, because this urea is very much soluble in water, and then it is less toxic. However, this ammonia that was present in the liver hepatocyte was very much toxic and insoluble in water. So due to this reason, whole these reaction takes place in liver, hepatocyte, cytosol and mitochondria to convert this ammonia that is reaches in the liver after catabolism of amino acid. So this ammonia finally will convert into urea through the urea cycle in your liver hepatocyte, and then this urea would be entered in the blood, and from the blood it will be reaches to the kidney, and from the kidney it will be excrete out from the your body in the form of urine through the renal tubule. So what happened? This ornithine, question is this here, what then happened with this ornithine? So ornithine again enter in this cycle. So that is the reason we are saying this chemical reaction, biochemical reaction, that it is a cycle and it is a urea cycle, because it is producing urea from the ammonia. And why it is cycle? Because the ornithine that is produced during this cycle will again combine another molecule of carbamyl phosphate that is generating from the from the condensation of another molecule of carbon dioxide and ammonia. So this ornithine that is producing at the end of this cycle will again combine with another molecule of carbamyl phosphate and then form citrulline, and then again citrulline would be will be going through the all these reaction and will form another molecule of urea and other molecule of ornithine. Then this ornithine again means utilized to produce citrulline. So this was about the urea cycle, how our body means how much unique this cycle is, and how means nature create different biochemical reaction in our body. So we could be means able to means get harm as much as means to the lesser extent. So here we will be talking about now a different rate limiting reaction. So what is the rate limiting reaction in this cycle? The first reaction that is condensing carbon dioxide with the ammonia is the rate limiting reaction in this cycle. What is the limiting reaction? Rate limiting reaction of a cycle or a pathway is that reaction that on that the means overall reaction of the cycle or a pathway depend upon. So in case of the urea cycle, all the reactions of this urea cycle would be depend on this first reaction, that is the condensation of these two molecule carbon dioxide and ammonia, and the and as a result of this condensation, formation of carbamyl phosphate. Because why it is the rate limiting reaction? Because until and unless this carbamyl phosphate will not form, so we cannot start this urea cycle. So this is the reason this reaction is a rate limiting reaction. A rate limiting reaction of a cycle is that reaction on that all other reaction of a cycle or a pathway would be based. So here what is the rate limiting reaction? Conversion of carbon dioxide and ammonia into carbamyl phosphate synthase, carbamyl phosphate, and the enzyme that catalyze the rate limiting reaction is the rate limiting enzyme. So what will be the rate limiting enzyme of the urea cycle? Enzyme carbamyl phosphate synthetase 1 would be the rate limiting enzyme of the urea cycle. So dear students, this was all about the urea cycle. So here I means give you all the text that I have already discussed you in the diagram. So for more information you can go through this link, and you can also read from there. So I hope you can understand what we have discussed here. For your more information I will conclude you how urea cycle work, and why it is needed for ammonia to convert into urea. Because ammonia, from first of all, from where we get ammonia in our body? We get ammonia in our body by oxidative deamination of amino acid. And from where we get amino acid in our body? From those protein that are metabolizing in our body. So what happened that when a protein metabolize, it generate amino acid in our body. So these amino acid would reaches in the liver and other part of our body, and especially in the liver oxidative deamination takes place. And after the oxidative deamination reaction, what happened? The amine group of ammonia remove, and this amine group, after series of chemical reaction, convert into ammonia. And ammonia is highly toxic and insoluble in water. So what happened that it would go in the urea cycle, and after the series of biochemical reaction, this ammonia would convert into the urea, and then urea would release from of our body from the kidney renal tubules in the less toxic form. So this was about whole lecture. I hope you would understand. Thank you very much. Allah Hafiz.